effects in single-cell RNA sequencing data. Here is the quick introduction and workflow for typical single-cell RNA sequencing data analysis, which is started from dissociating target cells from the tissue, cell isolation, RNA extraction, cDNA synthesis, and single-cell sequencing, and achieving expression profile. After quality control, normalization, feature selection, dimensionality reduction, and visualization, in some cases, scientists have to do further steps, which is called batch effect correction, before cell clustering and downstream analysis. What is batch effect in single-cell RNA sequencing data? Some systematic variation or biases introduced during the experimental or technical process that can confound the biological signals of interest. This variation often arises when single-cell RNA sequencing data are generated across multiple batches, such as different experimental days, instruments, or the experiment which is done by different lab personnel. Batch effects in single-cell RNA sequencing data can cause several problems, such as biases in gene expression profiles, incorrect cell type assignment, difficulties in distinguishing between true biological variation and technical artifacts, and create the illusion of entirely new cell types that do not exist in reality. Thus, by eliminating batch effects through data harmonization technique, researchers can uncover cell types with greater precision and confidence, ensuring the cell type assignments are based on genuine biological distinction rather than technical noise, and it helps them to discover new biologically relevant cell types.
In the second part of the video, we will talk about different algorithms for batch effect correction. When you want to compare different algorithms for batch effect correction, you can here choose all of the models that you want to compare, have them perform batch effect correction, and on the corrected data, run some different metrics or visual representations so that in the end you can rank them based on their performance in correcting batch effects. In this video, we will focus on SCVI tools model that, as you can see, rank among the best. SCVI tools is a Python package and a collection of latent space-based models for single-cell data analysis. A latent space is a lower dimensional representation of compressed data. These models can therefore perform some dimensionality reduction and removal of unwanted variation, such as correction of batch effect. These deep learning models are based on the variational autoencoder, which is a mathematical model in which the input data, here the matrix X, it can be an image, as in this example, or a gene count matrix in transcriptomics data, and in the SCVI tools use of the variational autoencoder. This input data goes through an encoder, which is a neural network of which the parameters need to be optimized. This encoded input is then the latent space, a compressed low-dimensional representation of the input. And then the latent space goes through a decoder, which is another neural network and with different parameters, which builds the reconstructed input, same dimensions as the input. The idea behind the variational autoencoder is that if the input and the reconstructed input are similar, then it means that you can compress your data into a lower dimensional latent space in which you have represented your biological variation and not the artifacts such as batch effects. So to quantify this similarity, then you can penalize the model by using the reconstruction loss, which measures the difference between the reconstructed input and the input. Once the model optimizes this loss, then you can say that you have optimized your latent space. In the probabilistic graphical model of SCVI, what is very noteworthy is that you condition the latent space on the batch ID of each cell, making the latent space unaware of the batch ID, and therefore the model performs some batch effect correction. The pipeline is that the raw input data is here, the raw count matrix that is represented here. And it's also conditioned on the batch ID matrix. So for example, here, this cell is from batch 0, whereas this cell is from batch 1. You will give all of this as input to a CVI and then train the model, choosing parameters such as the number of epochs or the different normalizations, or how you pre-process the data. And then when the training is over, then you can say that you have a optimized latent representation of the data, which can be very low dimensions, such as 10 dimensions over the genes. And on this latent representation, you are supposed to have gotten rid of batch effects, and you can perform some downstream analysis tasks such as harmonization, visualization, and clustering. This pipeline is easily adapted in code, and you can find it in the attached tutorial. Thank you for watching.